Hi everyone, I'm Dr. Lindsay Thiessen. I'm the extension plant pathologist covering field crops, tobacco, and now hemp. I wanted to take this opportunity to share some of our hemp foliar disease management work. So there's several different fungal leaf spots that can affect hemp, and these may not only impact just quality of plants, but they can also impact the yield by taking away leaf area that's needed to make the flower production or seed production or whatever portion of the plant that you're trying to harvest. So Cercospora leaf spot is commonly referred to as frog eye leaf spot because it has a darkened margin and a light center within the lesion, and that gives it that characteristic frog's eye look. One that's more local to North Carolina is called Exerohilum rostratum. It's a different leaf spot caused by a different pathogen. You can tell that it's a much darker lesion, a much more dark brown, and it still has that characteristic darkened margin, but you'll see a very distinct halo around each of those lesions. Another one that you might come across is Septoria leaf spot, and it is more of a creamy light brown with a yellow halo surrounding that leaf spot. All of these can be very easily confused in the field, so if you need a little extra diagnostic help, don't forget to make use of the plant disease and insect clinic here at NC State. So what can we do about all these different leaf spots? So there's some restrictions on what we can spray for as far as pesticides are concerned. For North Carolina, our rules are that you can spray things if they're on the minimum risk pesticide list, that 25B pesticides that are labeled for industrial hemp can be used in North Carolina. What that means is if the label is broadly labeled enough to include hemp and it's a minimum risk, that's something that can be used. I have an example here of a garlic oil product that has a broad enough label that says, but not limited to, as far as crops that it covers. Some other pesticides that have an EPA label as of this year, several of them are for insecticides. So there's some neem oils and other things. As far as fungicides, we have something like regalia, that's a Reinutria extract. There's Stargus, which is a bacillus species, or double nickel, which is another bacillus species strain. And those things now have an EPA label that can be used in hemp. So some cautions for pesticide use. Don't forget that the label is the law. We have to follow those state and federal regulations. So if you're interested in using a product, make sure you read the label carefully that you can use that. Any fungicide comes at a risk. Not only are residue limits not set in hemp because we haven't had this crop around long enough, sometimes the use can result in crop rejection from a buyer. So make sure that you're being in good communication with whomever may be buying the product, that they are aware of what chemicals you're using. We also are not sure about phytotoxicity impacts. So some of these products may impact plants in a negative way, which is why we do so much testing. So this year we have a foliar fungicide trial that's out at Central Crops Research Station in Clayton. This is just a picture of that trial. Most of our plants are just now at flowering, so we've started our fungicide program and are scouting now for foliar disease. Here's some of the products that we're using. We're looking at a lot of those products that have some labels. We're looking at Lifeguard, Double Nickel, Stargus, Regalia. We're using Rango, which is a neem oil product, which is typically considered an insecticide, but it may have some impacts if we get something like powdery mildew. But we're comparing everything to a conventional pesticide control, Quadris, which is azoxystrobin. Now that said, Quadris is not labeled for use in hemp, and I don't encourage anyone to use it. We're just using that out there to make sure that we have some sort of a positive control to compare to. So since we don't have any data from this year, I wanted to do a quick update from last year's trial. Last year's trial was a little different. We wanted to compare what chemistries may give us some efficacy. So we looked at conventional, organic, and low-risk pesticides, and we looked at the variety Bayox. So several of the products we used, we looked at metconazole, pyraclostrobin, a Revysol project, which has methventrofluconazole, pyraclostrobin, fluxapyroxad, and pyraclostrobin, which is preaxorzemium. So there's several different conventional products we looked at, and none of these are labeled. We also looked at several organic treatments. Again, several of these are not labeled for use in hemp, but they may have the potential in the future to be. So we looked at oxidate, serenade, serafel. Several of these are bacillus species that have some efficacy in other crops. We also looked at some others that are from Biosafe, Percarb, and Oxidate 2.0 with Oxyphos and Pvent following that. Then we looked at some lower risk treatments. We looked at a stylet oil, which has efficacy on powdery mildews. We looked at sulfur as microthiol disperse, and that one is also something commonly used for powdery mildew control. We also looked at a mineral oil and that alio product that I showed the label for, which is a garlic oil. We of course made sure that we had our nutrients out there. We did some weed control with cultivation and dual. And we also made sure that we had some late season foliar applications. It was a very dry year last year, so a lot of that is in response to some of what we were seeing in the field. So this is what our field looked like in July, about the same as what it does now. We were wet flowering at that point. As we get through the season, though, we noticed some tip dieback, and that was caused by a fungus that came in response to those drought symptoms. And with those drought symptoms, it caused that dieback, and it invaded the vascular tissue. We also had some fusarium wilt in our field. So you can see on the left what that looks like, where the plants have their vascular system collapse and then they just look dead. So our flower disease that we evaluated last year, our only treatment that reduced flower disease significantly was that microthiol disperse. Our predominant disease was that tip dieback pathogen that I was describing before. So we had microthiol disperse reduce the impact from that. Again, with our foliar disease, most of the lesions we saw were caused by that tip dieback pathogen. Again, the sulfur treatment, the microthiol disperse, reduced the level of disease in those plots. So from that, our only treatment that reduced the disease that we found was microthiol disperse. And what we're suspecting that happened was that we had sulfur deficiency due to drought conditions, and that increases susceptibility of those plants to the Lasiya diplodia pathogen. These plants received a sulfur treatment at transplant, but that may not have been enough to counteract our drought stress. We also had our foliar disease correlate with our stem disease, so we're pretty confident that what we were seeing in the field was a stress response. We had a lot of drought stress, and we did not have a whole lot of foliar leaf spots, so I think that's part of explaining what some of our symptoms were out in the field. So it looks like our Bayox variety was susceptible to that stem disease, but resistant to a lot of those common foliar pathogens. We took a look at our OVT plots last year and rated those for that leaf spot that was very prevalent in the last year. We had several plants that were so extremely sensitive that the leaves curled up and desiccated by how much they were affected. So there were several different varieties that had lower incidence. I have those circled in red: Bayox, Cherry Bee, Cherry Wine, but several were very sensitive. CO2, Midwestern, and Sweetened were all very sensitive to this Exorohilum leaf spot. Now that said, this set of varieties may be different from place to place and wherever you may get those clones. So if you're looking to use this as a guideline, take that into consideration that the plants that we have may not necessarily be the same genetics as another plant. So what does all this mean for our production? There's yield and quality effects from diseases. We're going to see reductions in yields and potentially our quality measures in plants that are affected heavily by foliar diseases. It's potentially capable of affecting our cannabinoid profile and can either go hot and increase our cannabinoid production, or in contrast, it may not produce enough. In our field trial last year, we saw a response be the opposite, where we had lower CBD and lower THC with those more heavily affected plants. Not only are we just reducing the yield from the direct impacts from the harvestable portion, but we can also see some outright deaths of plants, and that can also be a really big consideration for our management in the future. Another thing to think about is that it's very important to follow the fungicide labels, and not many things are labeled yet for hemp, but it's certainly an effort that we're working towards and trying to get more products available to growers. So don't forget, the label is the law. So thank you all for your attention. And if you have any more questions, you can follow us on Facebook or Twitter. Check out the hemp portal or our website. And none of this work would be possible without funding from NCDA, the North Carolina Biotech Center, support from the Plant Disease and Insect Clinic, and of course, NC State Extension.